A draft of a scientific article authored by Chinese researchers has drawn the attention of those in charge of bioRxiv, a database responsible for analyzing preprints in the biological field that will be submitted to specialized journals. This study involved the manipulation of a strain of the coronavirus, a species similar to SARS-CoV-2, which was responsible for the global COVID pandemic in recent years. The data released by the Chinese team has alarmed experts and science communicators worldwide, particularly due to the potential catastrophic implications. The high risks and very low potential benefits associated with such work are concerning. The preliminary results disclosed indicate a 100% mortality rate among the subjects involved in the project, along with an exceptionally high level of mutation and infection. This alone should be sufficient to halt any progress that could harm and threaten billions of people. In this video, we will share the details we have gathered so far and highlight the primary risks and conditions under which the research is being conducted. Stay with us to understand what is unfolding. The news became public in the third week of January through the British news portal Daily Mail, quickly gaining traction in major media outlets worldwide. As of now, the research seems to be in its early stages, with the results not officially published in scientific journals. However, it is already causing concern among most experts who fear the potential impacts the study could have, particularly given the conditions under which it is being conducted. The responsible team comprises approximately 10 professionals, with some reportedly affiliated with the country's military forces, according to the report from the news portal. The researchers cloned a strain of the coronavirus, belonging to the same family as the agent causing COVID-19. The distinguishing feature of this class of pathogens, which also gives them their name, is the protein crown forming their outer structure. This crown is responsible for the virus transmission, connecting to host cell receptors to infect them. Generally associated with respiratory diseases, coronaviruses typically cause nothing more than a common cold. However, with recent natural mutations and those conducted in laboratories, they have become an increasing concern. This experiment in Beijing involved cloning and inducing mutations in a virus found in pangolins, known as GXP2V. These mammals, resembling armadillos and anteaters, primarily inhabit Asia and Africa, posing no risk of infecting humans without our interference. The scientific community has raised questions about why manipulate a virus affecting these animals to make it transmissible. If the virus naturally found in these animals cannot be transmitted to humans, why artificially induce mutations in a laboratory to make it transmissible? This has become a point of contention and concern within the scientific community. What might be behind this, beyond scientific curiosity, is undeniably the daily advancement of science, bringing improvements to our lives through new technologies, the development of cures and treatments, and the generation of knowledge that allows us to better understand the past, present, and future. It expands our boundaries to unimaginable places, with even the sky no longer being our limit. However, every research endeavor, experiment, or any type of scientific investigation needs to be well established with clear parameters and objectives. This includes the implementation of safety mechanisms to protect those involved and, of course, transparency in methods, ensuring the fidelity of results and the possibility of study replication. As we will see shortly, this is not always the case. Firstly, the objectives of the research being conducted in China have not been disclosed, raising the first point of concern. After all, there are no answers to the questions of why, how, or for what purpose. Without these answers, analyzing the risks of this project becomes challenging, especially considering the sensitivity of the subject, particularly after all that we have recently faced. But it doesn't stop there. The methodology used by the team is quite conventional. 3 groups of mice were selected, each containing about 4 individuals subjected to different conditions. One group was exposed to the new mutant coronavirus, another received inactivated virus samples, and the last one was not exposed to anything, serving as a control group. However, there's something intriguing in this process. The chosen animals were not regular mice but humanized mice genetically modified to resemble humans more physiologically. While mice are typically used as research subjects due to their existing similarities to humans, in some cases, their body composition can be altered to further resemble humans. This ensures that research results are more reliable when compared to human subjects. In this case, the animals were prepared to have lungs similar to humans, which are the primary target of coronavirus infection. The researchers' intention is not merely to check the infection rate in other animals that are not natural hosts of the virus. If that were the case, there would be no need to induce laboratory changes to the original strain. In fact, their aim is to verify the transmission and infection rate in humans. Now, as the situation worsens, the pre-results described in the article to be submitted for publication indicate that 100% of the subjects exposed to the modified pathogen suffered within just a week. This implies that if this strain comes into contact with humans, the mortality rate is likely to be extremely high, equal to or surpassing what we have witnessed with COVID-19. The mechanism of action of GXP2V bears a striking resemblance to SARS-CoV-2, but it appears to be even more refined and adapted to progress towards severe cases. This was clearly observed in the test subjects that had exposure to the active virus, where the microorganism's proliferation rate was extremely high. It initiates in the upper respiratory tract, nasal cavity, larynx, and pharynx, and rapidly spreads to the lower respiratory tract, trachea, bronchi, and lungs, where it seems to settle and multiply more efficiently. While this follows the typical cycle of most respiratory disease-causing agents, these new coronavirus-based variants are becoming increasingly aggressive. In this case, GXP2V was observed to spread to the eyes and brain of the test subjects. This spread was identified as the primary cause of the high mortality rate among the subjects. Symptoms included fatigue, rapid weight loss, and whitening of the eyes. During monitoring, it was noted that the viral load increased significantly in the infected individuals. However, by the 6th day, the viral load in the upper respiratory tract decreased considerably, while in the brain, it continued to rise. After 1 or 2 days of this observation, the test subjects succumbed. Similar patterns were observed in analyses conducted with patients diagnosed with COVID-19, especially in cases involving the latest variants. Despite reports of neurological problems after the disease, such as difficulty concentrating and memory loss, this does not seem to have evolved as a critical factor representing a life-threatening risk. The complications arising from SARS-CoV-2 infection were present in other aspects. The concern lies precisely in the event that something more severe occurs. We could face something much greater and more deadly than what we experienced in the last pandemic. Towards the end of last year, documents obtained by the U.S. nonprofit organization, U.S. Right to Know, revealed funding requests for U.S. intelligence services to conduct genetic research on virus strains similar to those responsible for COVID-19. While this does not necessarily provide evidence that SARS-CoV-2 was deliberately produced in a laboratory and released into the environment to infect people, it raises concerning questions. These documents indicate that research is being conducted, either wholly or partially, through clandestine mechanisms aimed at circumventing established safety standards, precisely in place to protect the progress of these studies. This is likely done to reduce production costs, as investing in security is both expensive and labor-intensive. All signs suggest that we may be at the mercy of large companies that, in exchange for saving a few million, are putting the lives of billions at risk. It is not certain that this is the case with this particular research. However, the lack of clear information raises suspicions, especially given the traumas we recently endured and the indications pointing to human greed's capability of producing irresponsibility. The fact that China is prominently involved in both this case and the COVID-19 situation may be linked to a possible relaxation of safety parameters and oversight bodies that should monitor these studies. It's not necessarily these bodies conducting or these experiments being related to military research or biological weapons production. Nevertheless, the risks of conducting research blindly should not be minimized, and without proper control, the chances of a potential leak and contamination rise alarmingly. If this were the case, authorities might be alerted too late, and many lives could be lost. Let's hope this is not the scenario, and the full publication of the study clarifies our concerns. It might seem like a lot to ask, but it's never too late to trust in the altruism of humanity. I'll conclude here.